All right, this is Mr. Labonte. I'm going to be going over the concepts of nucleic acids and fats in this presentation. So we're continuing on with our four major biomolecules with a focus on the monomer-polymer relationships. And in that vein, the nucleotide is the monomer of the polymers DNA and RNA. So these are the polymers. Each monomer nucleotide is made up of three parts: a phosphate, a deoxyribose sugar, and a base. And we'll look at these in detail in the next few slides. Here's a more detailed image of a nucleotide. Please remember that every time you don't see a letter in these bonds, that's a carbon. And so what we want to point out first is we have a sugar here, but this is a 5-carbon sugar called a ribose. So the specific type is ribose here. These are, you may have heard, this is adenine, cytosine, guanine, thymine, and uracil. So these are the nitrogenous bases that are here, and then there's this phosphate group with a phosphorus on the inside. When we look at transcription translation, we'll talk more about the details of that. But for right now, again, this is just another look at the 1, 2, 3 parts of the. Okay, then here is the polymer. Two different versions. You can have RNA and DNA. RNA is in a single strand, DNA is in a double strand. But this would be the monomer, or one of these. Those would be the monomers. And then this whole thing would be a polymer, or that whole thing would be a polymer. So both of those are the polymers. Now we're going to take a more detailed look at the differences between RNA and DNA. And I want you to understand this because I want you to understand where the name comes from. And it starts really with understanding that the sugar that this is built on is called ribose. So ribose is this 5-carbon sugar. And normally, on the second carbon, remember our naming, our ordering sequence for carbons, normally on the second carbon we have an OH molecule there. However, in DNA, this second carbon, let's do this in red, does not have that OH molecule. It just has that H molecule. As a consequence of that, the name for DNA is deoxyribose. So that is, we've lost the oxygen. So, lost oxygen. And then nucleic acid is the last part. So deoxyribonucleic acid. So that's where this name comes from, deoxyribonucleic acid. Again, we'll look at all the pyrimidines and purines and bases when we get to looking at transcription and translation. But I want you to understand how this works with the ribose, okay? And that's it for nitrogenous bases. So far, we've gone through carbohydrates, we've done proteins, and now we've done nucleic acids. Nucleic acids consist of a monomer, which is the nucleic acid, and then the polymer called DNA and RNA. And then there are some specific examples, but you don't really need to know that. And now we're going to move on to fats, which is the final grouping. My favorite cartoon in the whole world, one of them. Just quickly, there's 3 primary functions for fats: storing energy, cushioning, which is quite important in the body. Like, think of yourself as running around and all of your organs bashing into each other. And then insulation. You can think of a big fat seal or walrus or something like that. Okay, actually, we're going to go back. Okay, so the first thing to focus on is fats are made up of fatty acids. And fatty acids are called acids because they have an acid component. Right here, you'll remember with amino acids and the carboxylic acid, that hydrogen can leave, becoming an H+, right? And that's going to lower the pH of a solution. So all of these are called stearic acid, acondinoic acid, linoleic acid, oleic acid. So we have all these acids. So if we're looking at the way fats are made, we have this acid part of the fat, and then we have this other part, okay? And I'm going to call this part the non-polar chain. And the reason I'm calling it a non-polar chain, it's important that you remember this, right? Maybe that red isn't the best. Because carbon and hydrogen have an equal electronegativity. So if they have an equal electronegativity, then there's not going to be any charge in this part of the molecule, and water will not be attracted to this part of the molecule. So it's important to note. So it doesn't matter how long this chain is. It can be really rather short. I think like 4, or the shortest I've seen are like 3 or 4, or it could be long, like up to 35 carbons. I don't really care. I mean, there's lots of different options, and this doesn't really matter for our purposes. But one thing you may notice is that sometimes you can have all of the carbons can be single bonded to each other and then bonded with 2 other hydrogens. That's the case here, and we would call that saturated. And so the idea is you just can't put any more hydrogens in this molecule. It's saturated. An unsaturated fatty acid would have a situation where you could put more hydrogens. So hopefully you see here that if you made that a single bond as opposed to a double bond, then you would need to add 2 more hydrogens to this molecule to make the carbons happy. So that would be called unsaturated, just like an unsaturated paper towel would have that option. But in an unsaturated bond, come on, I'm not erasing here, not very quickly, you would not have, you'd have double bonds in your carbons. So I'm trying to erase this so you can see those double bonds again. There we go. So there's double bonds in the carbons. We could add more hydrogens. This ends up having a huge impact on the molecules, and hopefully we'll have time to look at that a little bit in this class, but I can't guarantee it given the pace that we're moving. So we have saturated and unsaturated, and we have a monoglyceride. So up here on top here, monoglyceride, so 1 glycerol. And it can have a specific name just depending upon how many carbons it has and where the double bonds are or not. So this is what we make triglycerides out of, or fatty fats out of. Okay, now this is different from all of the things that we've talked about so far. So in all of the discussions that we've had so far, you have been able to add monomer to monomer to monomer to monomer to monomer forever, right? So you could keep adding monomers until the cows come home. And so you would go from a monomer to a polymer just by adding and adding and adding and adding and adding. That is not true of lipids. So I'm going to try to erase this. Let's see if I can erase these things more quickly. In lipids, the way that these fatty acids are added is they are added to another molecule. And so the molecule that they're added to is called glycerol. And what it's important that you understand about glycerol is there's 1, 2, 3 carbons in a glycerol. And you cannot add fatty acids anywhere except to the oxygen that is connected to one of these 3 carbons. So you could add 1 fatty acid chain here. You could add, hopefully this won't be too ugly. You could add another fatty acid chain here. And you could add a 3rd fatty acid chain here. So you can add, this is 1 fatty acid chain, 2 fatty acid chains, 3 fatty acid chains. So let me see if I can undo all this to make it so not so ugly. This, what we're looking at right here, is called a monoglyceride. And the reason we're going to call this a monoglyceride is we only have 1 fatty acid chain that's connected to it, right? I'm going to go to the next slide and you're going to see a triglyceride. So the organization can be a monoglyceride, which is 1, a diglyceride, which would be 2 fatty acid chains, or a triglyceride, which would be 3 fatty acid chains. So I'll review that on the next. Okay, so here what we see is a triglyceride. So there's our glycerol molecule. This is 1 fatty acid chain, 2 fatty acid chains, 3 fatty acid chains. And because there's 3, we call this a triglyceride. So a triglyceride. Now this looks a little bit different. Remember, these are all carbons, and these would actually have hydrogens on them as well. This is a really shorthand description. You don't need to know anything about where these double bonds are, but hopefully you can see that this one is unsaturated. That is, all of the carbon bonds have hydrogens. And sorry about that. Let me try that again. This one is saturated. All of the double bonds have hydrogens, or all of the carbon bonds have hydrogens, and these 2 are unsaturated. And what unsaturated means here is you could add additional hydrogens to this molecule. All right. DIFF. Oh, sorry about that. We're probably going to, I'll talk through this. You may not need to know this. In situations where you have unsaturated bonds, so you see these are unsaturated, right? So in a situation where you have an unsaturated bond, you can have 2 options. One option is that both hydrogens are pointing up in the same direction. You see that? The other option is that 1 hydrogen is pointing up, 1 hydrogen is pointing down. Cis means same, and trans means different. Different. This matters quite a lot in biology because this orientation, and I'm going to highlighting green now. This orientation does not exist in nature. So this is not an orientation that you would see in any plants or any organisms. This is the orientation that exists in nature, the cis. So I'm going to write natural here and unnatural. And your question might be, well, if it doesn't exist in nature, how do we know about it? And the answer to that is we know about it because humans have made it. So what we can do is we will take an unsaturated fatty acid. So we may have what's called polyunsaturated. So there'll be a bunch of double bonds in here. We'll take these fatty acids, we'll put them in a big old vat of fluid, and we'll bubble gaseous hydrogen through the fluid, and that will saturate these double bonds, and when it saturates these double bonds, it will make these trans bonds here. So have you ever heard of hydrogenated vegetable oil? If you look in peanut butter, you will see hydrogenated vegetable oil. And what that means is they have taken vegetable oil and they have heated it up and bubbled hydrogen through it. When that happens, you will get something called a trans fat, an unnatural fat. And yours truly ate a lot of trans fats when he was a kid because I loved peanut butter, and nobody knew that trans fats were not so awesome. But what has happened in the intervening years is we have realized that these molecules, this whole molecule will go into your body, and it will be incorporated into your body. And when it's this molecule that's incorporated into your body, you're fine. But when it's this molecule that's incorporated into your body, it's associated with a higher rate of cancer. So that's why we're avoiding trans fats, if you've ever heard of trans fats. Yep, and so this is just what I just said. This is the side effect of chemical hydrogenation, is you get these trans fats. This is what you would like. You would like complete hydrogenation, so you get a completely saturated thing. And so the way people are avoiding this is probably you have seen in your lifetime all of this excitement about coconut oil and coconut fats and avocados and things like that. So these are oils that are solid at room temperature. So they have a lot of these traits that we like, but they don't have any of these things. Solid vegetable oil oftentimes are used to have a bunch of trans fats. Peanut butter, a lot of things like things that say they don't have animal products, but they have solid oils in them, like Hostess cupcakes and things like that. That's where you're likely to get these trans fats. And we're going to skip. We're going to skip the linoleic and oleic acid. But I want to talk a little bit about absorption and how this works. Because fat is, as you got at one point, I just said these fatty acid chains are absorbed. And that's a difficult thing because these are big molecules compared to sugars. So let's say you're eating something, all right? You might have a triglyceride. So I'll do this, dot, dot, dot. This is my drawing for a triglyceride with my 3 fatty acid chains. The acid in your stomach can break this down to a diglyceride. Ugh, that was ugly. So the acid in your stomach might break that down to a diglyceride. That's my drawing for a diglyceride. But once this happens, what's going to happen when you have a diglyceride is you're going to form this thing called a micelle. okay? Sorry, and then you're going to actually have, prior to that, you're going to add bile, and that's a salt. And the bile salt is going to bond with this diglyceride to make a micelle. So the green here is bile. And then these yellow lines, these are the fatty acid chains, okay? And bile is polar. And these fatty acid chains, as you should remember, are non-polar. So what happens is all of the polar parts want to connect to the water outside, and all of the non-polar parts want to hide or go away from the water outside. And that's what gives you this micelle. And then once that happens, these micelles, because they have these special little bile salts sitting on the outside, that allows them to be absorbed into the bloodstream. And then we'll talk a little bit about how they go in through the circulatory system. But the way that they go into the circulatory system, they get surrounded by some proteins. So you're going to have proteins and these fats, and lipo, and that makes a lipoprotein. And that image that I've included, there's one image in this week's work. That's an image of a lipoprotein. And that's what we're going to be looking at for the next lab. This is just a bunch of different fatty acids and their saturation content. All right, the last thing I want to talk about is the magic of soap, okay? Now, to start with, I want you to get that in a lipid, so I think I might have, in a lipid, and I'm just going to draw a triglyceride. We have a glycerol, and then we have a fatty acid. So we have carbon, carbon, carbon there. Okay, I'm going to have to erase this. Try to get this color coordinated here. I think I'll have my carbons back. Carbon, carbon, carbon. Oxygen, oxygen, oxygen. And then carbon, carbon, carbon. And I'm too lazy to draw them, right? And all these carbons going out. And then we have hydrogens all over the place here, right? So what is very important for you to understand about this diagram is everywhere I'm highlighting it, right? All of this area is non-polar. This means it does not like water or is not attracted to water. Water does not want to deal with this molecule. This is why it's very hard to wash oil off of a pan when you don't have soap, because water doesn't like nonpolar substances, and nonpolar substances don't like water. In fact, nonpolar substances are called hydrophobic or water-fearing, right? Phobia is a fear. So nonpolar substances are called hydrophobic. So what is so magical about soap? Sometimes I call soap like the perfect marriage counselor or something like that. So if we look over here at a soap molecule, we see this is just basically a bunch of carbons, and it doesn't state it on here, but a bunch of hydrogens. So this whole part here is nonpolar. So nonpolar things are going to be attracted to nonpolar things, right? So this orange part here is going to be attracted to this orange part here. However, take a look at this over here. Do you see this charge? Well, when we have a charge, we can safely assume that we have a very polar situation, right? So this negative oxygen is going to be very polar, and it's actually going to be attracted to the positive hydrogens of water. That's my oxygen. Here's my water molecule. And there's going to be an attraction between the positive hydrogens and the negative oxygen right here. And so what happens with soap is, and I'm running out of space here a little bit. Let's move this over, and I'll draw it right over here. What happens with soap is you're going to have let's pretend we have a pan. And in this pan, we're going to have fats. I'll denote a fat by doing the glycerol and the 3 lines. So this looks like an E, right? But in that E, what I'm saying is that this is the glycerol part, and then these lines are the fatty acids, okay? So we have these little E's, okay? And now we're going to have soap, and I'll draw soap in blue, okay? Soap is going to have 2 parts. It's going to have the nonpolar part. That's that blue line. So that's this part right there. That's the nonpolar part. And then it's going to have that red oxygen, that polar part right there. And I'll put a minus sign there. So that's this part right there. Okay. And then what's going to happen, because I'm running out of colors, I'm just going to do this in green, is we're going to have a water molecule. And that water molecule, maybe here's your sink, right, is going to be coming down like this and falling. And you're going to end up with a connection between these 3 things, right? So there's a hydrogen. There's this attraction between the hydrogen, the positive hydrogen, and the negative part of the soap. And then there's also an attraction between the nonpolar part of the soap and the lipid, or the fat molecule, because they're the same. So the water molecule leaves, it pulls this part of the soap molecule, that's A, it pulls B, and then that part of the soap molecule pulls the fat, or the lipid, C. So that is how we wash dishes. And that's it for me.